Welcome, everyone. Thanks for coming. Um, my name is Trevor. I'm a PhD student in the department. I work, I think, directly below us, a couple floors down. And today I was invited to share a project from early in my PhD about web-based visualization of biomedical imaging data. And in particular, I want to talk about sort of how a convergence on how cloud-based formats really, at the same time as developing this web-based visualization, led to alleviating some common bottlenecks in bioimaging analysis. So for context, at the time when we first started this project, the Gellenborg Lab is a part of the HubMap consortia effort, and there are a growing number of technologies which measure single-cell data in human and other tissues. And a primary goal of some of these atlases, or some of these consortia, are to build these comprehensive tissue atlases, which integrate omics data sets, including related sequencing, mass spectrometry, and high-resolution microscopy data. And so as a part of the Gellenborg Lab, we were tasked with, as on a larger team within HubMap, to build out infrastructure for the public data portals. And in particular, these data portals are very important entry points for scientists and individuals to preview and explore these growing tissue atlases. And so we have to think a lot about the visualization, but also about the infrastructure that can support browsing these growing data sets. And so, as I mentioned today, I'm going to talk about one very specific aspect of this work, and that was focusing on high-resolution microscopy data, and a project that spun out in order to support that high-resolution microscopy data within HubMap, and then also the development of cloud-based imaging formats and open standards. So an early question within this process of designing these visualization tools for the data portal were thinking about what imaging format made sense for us to choose, or was an imaging format the right way to think about this problem at all? And so we quickly went into a deep dive into the biological imaging field, and it is rapidly evolving, and there are a proliferation of technologies without really a clear, suitable, interoperable standard. And so this has led to many, many, many proprietary file formats. And at the time when we were evaluating these different technologies, there was no clear choice for a particular format that was both fair but also flexible enough for the new types of data types and data structures that were coming from the various modalities in modern biomedical imaging. So just at a very high level, I just put this figure of a bioimaging data set, which includes both sort of acquisition metadata about the particular image, and then a big component, and probably the majority of the size of these data sets are due to the binary pixel representation. And so that's sort of consistent across all these different formats, but they all choose to represent these data differently. And then I've sort of put this checkbox at the bottom, which indicates, I think, a growing need to have a format which is flexible enough to integrate or express linked modalities with imaging data that they themselves may not be image-based. So the current mechanism of providing sort of this interoperable layer when we first were evaluating these tools, or sort of the status quo, is found in the form of translation and conversion of these proprietary file formats. And so what that means is there's a particular tool that's very important in the field called bioformats, which is responsible for this translation and conversion of these proprietary file formats into open standard protocols or open standard formats. So when I refer to conversion, that's really the right side of this figure, which is showing sort of a permanent path, which is a part of this bioinformatics or bioformats process, which converts some vendor format in a two-step workflow into first a multi-chunked format, and then last a pyramidal OMI-TIFF open standard format. And conversion means that it's a permanent copy of that data set. The other method for dealing with interoperability is found in the form of on-the-fly translation, and that's shown here on the left. And what that connotes is that bioformats is sort of a persistent process which can be used in applications to repeatedly create transient interoperable representations of the data. And so as you can see, bioformats has a lot that it takes on in order to sort of accomplish this like compatibility layer. And on-the-fly translation is this active process for some applications like Omero for keeping these data interoperable with various tools. So how does this all work with bioformats? And the answer is really that it's a really large open source project that takes a lot of maintenance in order to support, I believe it's over 150 different vendor formats to keep up to date with both the latest formats and then also some of their derivatives as well. However, I do think it's worth noting that there are common patterns that are found across all these formats, which is why bioformats can sort of accomplish this task at all of converting these and representing them. And in particular, I've listed some attributes here at the binary level of how these data are represented commonly across these different formats. So data are typically chunked. That means that the large X and Y planes are typically split up into smaller tiles so that they can be accessed individually from disk. Each of those tiles or chunks are compressed so that the file on disk takes up less space. And then data are often already pyramidal. And so when I say pyramidal, I've included this figure here on the right. This means that often when the images are initially captured at a very high resolution, and far more pixels are binary pixels are in that high resolution than there are on any screen that you might have. And so in order to load that whole image into memory would take a lot of memory. And so rather than doing that, there's a method of pre-processing the data that downsamples the individual high resolution image so that it can be accessed at several different levels. And what that allows you to do is it's sort of like a Google Maps view of looking at your microscopy data. If you look at the very top of the pyramid, you have like almost a very small tile of your image, and as you zoom in, you can progressively load just the parts of the image that are important to your region of view, and it allows for very efficient browsing of these data sets. So there are limitations to this translation and conversion methodology, and in particular, translation is limited in terms of scalability. And on-demand translation is an expensive bottleneck for large data resources. So resources which have like terabytes and petabytes of data to constantly be translating those on the fly can be quite expensive for and bottlenecks in these large data pipelines. And even moreover, many of those vendor formats which are supported by bioformats are designed for acquisition, meaning that they're optimized for write and not read. And so as you're reading from those files, if it's a write optimized file, there's somewhat of an overhead, or it's not meant to be accessed in that type of way. And so translation sort of is this limited in terms of its scalability, and it does not sort of grow for these larger data resources. Now on the side of the conversion, open standard formats are somewhat lacking. So OMI-TIFF is a sort of the de facto open standard in the field. However, it's designed off of, it's built on top of the binary representation as TIFF, which is fundamentally a two-dimensional format. And so for these new data sources, which are might have large 3D volumes, OMI-TIFF isn't really a suitable format for representing those data. So another open alternative might be HDF5, which allows for sort of these multi-dimensional representations of the binary data. However, both HDF5 and OMI-TIFF are both these sort of large binary files which do not allow for efficient cloud access. And so while they're well suited for reading from disk, reading these over a network connection can impose quite a high overhead. And then finally, neither of these really have a specification for linking non-imaging modalities. And so, in summary, the conversion of OMI-TIFF or to OMI-TIFF or HTF5 requires users to download entire data sets. So because these are not cloud optimized, prior to analysis or visualization, users are forced to download these data sets wholesale before performing either visualization or analysis locally. And translation is a very expensive step and can be a serious bottleneck at scale. And this essentially prompts the need for a more efficient remote read access format that allows sort of these streaming data access patterns by end users. So I did hesitate to, there's that famous XKCD quote about creating a new standard, and I almost put it in, but I decided not to because I'm hoping to convince you that it's not just another competing standard, and that in fact, the project that I worked on in combination with some of these visualization efforts was this design of a next generation file format, or the OMI NGFF format, which is a new community standard and has an open specification, which is meant to be to sort of complement the OMI-TIFF and the other and HTF5, which are the other sort of open data models. With the goal of satisfying a lot of these issues in bioimaging, and specifically bioimaging at scale and sharing of data. And OMI NGFF is built on top of the ZAR format. So you might hear me say OMI ZAR throughout this presentation. The two, for the sake of this presentation, are interchangeable. And a very important aspect about this format is that it can be stored natively in cloud storage. And thus it opens up direct access to many more users. So an important part of the picture of that painted by the OMI community about these different formats is that really they're complementary. So OMI-TIFF, HTF5, and ZAR NGFF are sort of these equivalent, they share equivalent metadata. However, the binary representation might be different. And so for those reasons, then these formats might be able to suit very different needs in the bioimaging community combined. And in particular, though, today I'm going to be talking about the advantages of using this new NGFF format. So not to go too far down the rabbit hole, but I do feel like it's worth introducing sort of at a high level what even is ZAR and how does it differ from these different formats. So on the tin, ZAR is a flexible format for storing chunked, compressed, multi-dimensional arrays. And a very important aspect of ZAR is that it's also, so it describes a flexible format, and then many different languages implement libraries for reading and writing that format. And so I've just displayed some of those languages here. I'm the lead maintainer on the JavaScript implementation, but it's a very wide community. And there's also a committee of voting on new features to add to the specification. And there's a lot of buy-in now from a much wider community than just the bioimaging community. So at a high level, an array, or sort of these binary data that we talk about, are a container of like items. And in particular, these various items within these containers are all the same data type and size in bits. And then the dimensions of these containers specify the number of items, and that's they're described by their shape. And so you might be wondering, like, why am I going down this rabbit hole describing the n-dimensional arrays? And that's because biological imaging pixel data are essentially multi-dimensional arrays. And so I've just drawn a little figure here of showing sort of a diagram of a multiplex fluorescence image with several different channels captured in an individual 2D plane, and then we can stack those planes together into a single 3-dimensional image. So it's a 3D array that has 3 separate channels and all the same X and Y. So why would you even need ZAR at all to represent something like this? And really ZAR comes into play when you have data that are small like this. This is very easy to translate or load into memory, but ZAR becomes a really big deal when suddenly your data are too big to fit into memory. And so the question is, If you had a plane this big and you need to load this on your computer, there need to be sort of mechanisms that allow you to process this, not all at once. And so ZAR is really a pipeline for sort of tackling this problem of how to break down these very large arrays into smaller chunks that that can be processed independently. So the first step of ZAR is to divide your array into chunks. So you have this large plane, and you sort of subdivide this into smaller pieces that we call chunks. And then each chunk is compressed. So that means you run some compression algorithm on the binary data, which allows this to be stored more efficiently on disk. And then when you read it back from disk, the chunk of data is decompressed. And a very important part about ZAR is that each of these chunks are given their own unique identifier or a key. And so in contrast to some of these larger imaging formats, a ZAR data set can be stored in any sort of key value store rather than just like a single binary file. And then once you have these separated chunks, when you then want to access the data, as shown here on the left, you only need to read that part of the data that's been written to disk and not all of the data at once. The conceptual model for ZAR is that it also allows, it's not only the binary data, but it also allows for sort of grouping of these larger data sets. So you could have multiple images within a single store object. And so ZAR is organized in sort of this hierarchical form. And the one piece that I want to point out again is that the chunk data, which are shown here in purple, are uniquely identifiable and can be accessed directly by their key name. And so this allows for me as the end user to load the chunks that I want directly without needing to read additional metadata about the store or the file. So this is a nice figure that was provided by Henning Falk for the ZAR community, which I think really breaks down how ZAR differs from these different monolithic formats. So, and it also contextualizes it in the form of bioimaging data, which is nice. So on the upper left, we have some 3D volume that is imaged and then split up into these separate, sort of like a Z stack of the separate 2D planes that make up that 3D volume. And then there's one of two routes that these data can be converted to, and the right being or the left being the monolithic file format, which is sort of a catch-all for HDF5 or TIFF, and OMI-ZAR, which is shown on the left, on the right. And so in the monolithic format, the important thing to note is that anytime data needs to be accessed, there's a read and seek process that happens. So data are read. A client reads from the header and then must find which part of the file to then go to then read those bytes into memory. Versus with OMI-ZAR, the array metadata is read once up top, and then anytime that you need to access the individual chunks, you can do so directly. And so a key piece here being that this file is, in this example, is 120 gigabytes, and this sort of directory or object store is made up of much smaller pieces that can independently be accessed directly. So in the original NGFF paper, we benchmarked how ZAR improves the remote access efficiency of these compared to these existing formats. So the y-axis here are different formats, and the x-axis are seconds per chunk. And the asset, or sort of the benchmark that we were running in this was to assess the time at which it took to read individual chunks from disk for different environments. One being a local file system, HTTP, and S3. And the piece at the top here shows the overhead of just reading a single byte from any of these environments. And so what you can see is reading a single byte from the local file system is super fast versus anytime you're going over a network, it's orders of magnitude slower to just read a single individual byte. And the important takeaway here about ZAR compared to TIFF and HDF5 is that the orange bump, which is HTTP, and I guess S3 as well, have much more efficient reads over the network. And this is really illustrated by the previous illustration, because over the network, you no longer have to both read the header and seek. You can access these chunks directly. And so ZAR sort of poses this trade-off, or sort of serves as a better cloud-friendly option for these data. I do think it's important to note that HDF5 locally is super performant, but it is sort of these trade-offs of both a read and like local read and remote read choice that I think are compelling about ZAR in this context. So the top here is for a 3D image that is 65,000 by 65,000 with 8 channels. And the bottom here is a 4-dimensional image that is a volume across time. And the main takeaway from this image is that we actually couldn't run this benchmark with TIFF because it's not, there was a memory error due to trying to load the amount of memory to represent these long, big planes with the TIFF format. And then it's just reiterating the observation from above that even for these volumes, ZAR seems to have a good improve this remote read access over some of these monolithic formats. So bringing this now into the context of the work that I have been doing in the Gellenborg lab, as we were deciding on these different imaging formats, I was involved in the design of the NGFF specification, but also in tandem with trying to build visualizations to support these open standard formats in a more direct manner on the web. During this process, the HubMap Consortium adopted OME-TIFF as sort of the open standard for which they share their data within the consortium. And they're committed to using OME-TIFF and adapting NGFF over time. And this forced us to sort of evaluate the current infrastructure to support visualizing these various data formats on the web, specifically in the context of the data portal that visualizations, which we'd eventually integrate into. So I just want to highlight at a high level the challenge of like drawing these pixels that end up on your screen. And the reality is that we have these large multi-dimensional images that contain many different channels that fundamentally those channels need to be mixed somehow and rendered into a RGBA picture or pixel that ends up on your screen. And so the way that I've denoted this in the picture is the blue tile represents what I call a data tile, and that's the raw data from the actual binary file. And at some point that has to be converted into this like RGBA tile that fits on your screen. And the mechanisms to do that when we were sort of surveying existing technologies, were all server-based. And we broke them down sort of into 2 primary categories of server-based architectures. So the first architecture is something that we called offline server-side rendering. And so the method of offline server-side rendering is very simple. Essentially, prior to deploying said web application, you create an entire copy of your imaging data set, which will not be used for analysis, but only to render it to a particular pre-rendered to imaging formats that are well understood by the browser. So effectively, you have your multi-channel microscopy image, and each of the chunks from that image you render into individual tiles that are either JPEGs or PNGs. And then this pre-render step creates this sort of folder that you can easily put in cloud storage or something, and the web client can easily request these data. The challenge here and the real limitation is that there's no way for the user of the web application to re-render the data. So any type of transformations that have been decided all have to happen in this pre-rendering step. So we found that these types of applications often fell into a category of explanatory visualizations, where scientists who had a very compelling story about their data that wanted to show particular channels turned on and off, or regions of interest, and maybe have like a guided view of a large microscopy image, would choose something like this because they can sort of pick the channels that they want to show, maybe render out several different image pyramids, and then have a very simple application. However, this isn't really a good fit for the types of exploratory tools that we're building as a part of HubMap, because we want people to be able to explore these growing data resources, and no person would be able to sort of curate these pre-computed visualizations. The next architecture that we surveyed, we defined as online server-side rendering. And in this application, you actually have both a persistent server process running alongside a web application. And the server is really like a custom architecture that is coupled quite tightly to the web front end. And this is because it's sort of doing this very complicated process of relaying what the user interface is desiring for a particular visualization, and then recreating that on the server, and then sending back that data to the client as a rendered image tile. And so this ends up having quite a complicated server architecture, and just to support communication with the web client, but then also to support this process of rendering the actual source images. And a very important piece of this type of visualization is that every user interaction requires a full round trip to the server to see any changes. So if I adjust like contrast settings or change a particular channel in the image, that from the web browser, I will need to issue a request to the server to then render that and send it back to me. And so there are long latencies involved in actually seeing the rendering cycle, because the rendering is happening elsewhere. So as we surveyed these various approaches, again, offline server-side rendering shown on top and online server-side rendering shown on the bottom, we saw that they sort of fit on this spectrum of where the primary image data is ultimately converted to an image tile. And we denoted this as DT, as data transform. So it's some sort of data transformation that takes your imaging data and then converts it to an image tile that is then ultimately sent to the web application. I think an important caveat to note here is that the primary imaging data is either not available or requires a separate API to access under this model. So the web view or the client view views these rendered tiles, but you see that there's no direct arrow from the primary imaging data to the client, which means that some other API needs to be exposed in order to support querying the actual primary data. And then finally, just a note about when we were looking at the front-end implementations of many of these different tools, we found that many of these were designed in an era where the web browser didn't have many capabilities that it now does, and in specifically graphics card accelerated rendering. And so many of these technologies are built off of very simple front-end tools, and we found that we really think that the tendency to work towards these client-based sort of these pre-rendered applications is that the imaging community is quite used to already converting their data for various compatibility reasons. And then second, the web technologies in the front end were suitable for loading these types of data. And so we introduced a new method in a tool called Viv, which is a multi-scale microscopy visualization toolkit for the web. And Viv does something special in comparison to these other tools in that it implements something called client-side rendering. And so in client-side rendering, between the primary imaging data being rendered to the final display, you can see that the actual data tile that I mentioned earlier is requested by the client. And so you can think of the web browser as acting how a desktop viewer would load data to view. Instead, the web browser is doing that and performing the rendering on the client on that GPU accelerated hardware. Additionally, it supports multi-scale two-dimensional rendering, some special features, and I'll touch on those in a second.
Maybe one very
yes.
understanding question here. DT, somebody in the audience asked, stands for data title. Is that true?
Sorry, data transformation. So it's denoting the transformation from the primary data to some rendering of that data. And so here the data transformation is executed on the client versus on the two other examples, the data transformation or this rendering is converted, is executed on the server. Great question. So Viv is really a JavaScript library that we designed to support these types of high resolution microscopy imaging data sets on the web. And really we built it because when we surveyed the different technologies that existed, we didn't have something that we could just plug into these open standard formats to be able to visualize them. And in particular, we built in support for the open standard formats from the open microscopy environment, including OMI-TIFF and OMI-NGFF. And we built it with these modern web technologies. And I'll show you some of the features in a second of how we leverage some of these technologies to do some pretty neat rendering types of things on anything from a mobile device to your web browser on your computer. So the first piece is, as I mentioned before, is that these data sets are many times, these images are already pyramidal. And so the data are designed to be accessed in this lazy manner from the client. And so we built in support for rendering these pyramids for both OMI-TIFF and the OMI-ZAR data formats directly in the web browser. So this is just a little preview of the multi-scale functionality. The next bit is that, as I mentioned before, that these multiplexed imaging data sets combine many grayscale single channel images together. And so a type of rendering that's often done is a pseudo coloring or pseudo mixing of these, where you apply a color map to an individual channel, and then those are all blended together. And because we do this within the client, you can see that here in the control panel, I can select different colors to assign to different channels and adjust these the different contrast settings for the individual channels. And the updates are applied very quickly on the user GPU because it's all being done on the client, and this doesn't take some sort of round trip to execute. Another feature is that we support the layering of these different imaging data sets together. So here's showing two different imaging data sets, one an imaging mass spectrometry image and another from a multiplex immunofluorescent microscopy image, which is multi-scale. And then we support affine transformations to align these two together and allow them to be visualized simultaneously in the web browser. Another feature, just showing some more of these rendering features, is the ability to, now that you have these two images layered together, you can actually adjust the opacity and blending, and that all happens seamlessly on the client because, again, we're doing the rendering on the GPU. We have some preliminary support for non-multi-scale 3D volumes. So this is showing a 3D rendering or a 3D raycasting for a simple 3D data set. And then finally, we allow for extensions to this rendering pipeline. So this is something that we introduced in our paper that we called magic lenses, which is this ability to pick a particular channel and highlight that channel directly under the lens, sort of blocking out the others. And the only reason we're able to do this so efficiently is because, again, that we're doing the rendering on the client. So you wouldn't be able to accomplish a feature like this if you had to have a server round trip, because the mouse is moving several times per second and you have to keep up with the rendering loop. And submitting multiple server requests for that just wouldn't work. So a very high overview of how Viv works is that it just inherits from this traditional pipeline that we outlined before. So we explicitly aim to not invent any new formats and to try to target these open standard formats that are defined by the open microscopy environment, so that they serve as these cloud-friendly formats for us to power these visualizations. And then you can directly upload these files to some sort of web server or cloud object storage. And then our library sits right on top of this on the web browser, and we'll load these data and perform rendering as these different layered components. Probably slightly a technical note, but we extend from this geospatial library called DecGL, and Viv is really like a first-class citizen within that component library. So you can combine Viv's imaging layers with anything from DecGL ecosystem to build much more abstract applications that combine different modalities other than just images. And for an example, we've built several tools in our lab which leverage this sort of composability of Viv. And one in particular that we're very excited about is called Vitesse, which is a web-based viewer for spatial single-cell data. And it, in the spatial view here shown, behind all of that, you can kind of see there's an imaging view. It's a bit blown out on this screen. But overlaid on top, we're able to highlight individual RNA molecules from a spatial RNA-seq data set. And we've been able to integrate this viewer within the HubMap data portal. And the test allows this like linked exploration of these multi-omics data sets with imaging data. Another use case, we built a tool called Vizar, which is really during this moment of trying to flesh out the OMI and GFF like compatibility. So Vizar is scoped sort of only for support for the NGFF specification, but it has some additional features that sort of show how these web-based views can integrate into computational workflows. So in particular, Vizar allows for integrates directly into Jupyter environments, which allows for the ability to visualize data on remote servers directly in your web browser. And so this is we in our manuscript describe a workflow, a novel workflow where a very memory-intensive imaging alignment occurs on a remote high-performance cluster, and within the same Jupyter notebook, you can we're able to inspect the registration results in the same notebook as which we ran the various registration. And this just simplifies the typical workflows of needing to switch applications or somehow find a way to granularly check these alignments when performing these types of registration. Vizar also doubles as a standalone image client. So any one of these formats, you can point to the simple cloud storage and then give that URL directly to Vizar, and it'll render it directly within your web browser. And the important thing to note here is that this is a link for the actual data set. So, and if it was a TIFF link, and you type that into your browser, you'd start downloading some massive image. And so it's a really powerful way we found to sort of deconvolute like where these data sets actually lie. If your cloud architecture supports this type of random access, then people can start sharing data sets via links rather than sort of links that then go to another page that then you eventually find like the data set in question. Okay, I have a couple more slides and then we should be good for questions. One thing I wanted to note is that earlier I talked about how OMI-TIFF sort of has these limitations in terms of remote access. And as a part of this, because we were looking to support OMI-TIFF as an open standard format, we looked for mechanisms for ways that we could try to sort of append on cloud optimization to a format like OMI-TIFF. And so similar to some methods that are actually used in the genomics community for like indexing different types of genomics data formats, we propose a indexing strategy for OMI-TIFF to improve its remote read efficiency. So the figure is showing like a cloud optimized OMI-ZAR. OMI-TIFF is not natively cloud optimized, and then our extension to OMI-TIFF is OMI-TIFF plus this small index file that allows you to read it more closely to how performance of OMI-ZAR. And we benchmarked this. So these kind of are similar to the figures from before. I'll walk you through them quickly. But here we have the different formats, and these are all looking at chunk times for HTTP requests. And what you'll see is that for an image that doesn't have very many channels or Z or T dimensions, the formats are somewhat similar. and that the benefits of using index TIFF don't really outweigh just using TIFF. But as the Z dimension grows, or as you have more seeking to do in that file, index TIFF very much outperforms the using native TIFF. And the final piece over here is just showing we were trying to figure out like what this dependency was, or on these different dimensions. So effectively, this plot here is this first plot, except we've also added the Z index as the y-axis. And so what you can see is that the Z index for the image. And so each point here is a chunk that was requested, and its alignment here means that it was requested from a different Z index within the image. And what you can see is that only for TIFF, there's this relationship between the dimensions and the chunk times. And so what we effectively are showing is that our index prevents that sort of relationship or sort of skewedness from there being this extended seeking in the file. When the original publication for OmiZar was first published, there were 3 accompanying tools that came with it. One from Java, one from Python, Napari, and then one from JavaScript, or from our project Viv, and that was Vizar. And there's been just sort of a growing number of these projects over time. But it was sort of this proof of concept about 1.5 years ago to just show that we could a data curation center like the Imaging Data Resource could release these large multiplexed images, and then all these different clients could be able to access those efficiently. And then since then, our tools have been integrated in various different environments to sort of allow these more cloud-optimized data access workflows. So just on the final leaving note, I just wanted to say that the number of tools that are, although there were those original 3, the number of tools around this next generation format are really growing. And it's much less of a experimental like area now, and it seems like it's sort of the accepted future of the field for processing these very large data sets. And these new tools include viewers, readers and writers, metadata converters. There's a large ecosystem now that wasn't there 1 year ago. And the current ecosystem is probably suitable now for solving many common scalable issues in the bioimaging community. And then in terms of the visualization, the integrated web-based viewers allow these sort of new workflows that we're still exploring in my research to both share and provide web-based views to these computational backends that typically weren't possible before. And the Viv tool has been adopted by other consortia outside of HubMap, including the Kidney Precision Medicine Project, as well as the Galaxy Project. And with that, I want to thank both my advisor and the folks from my lab that worked on this project, our collaborators within the HubMap consortium, my funders, and then finally the folks from the OMI community who really welcomed my involvement in the format, particular Josh Moore. And I think it really led to an exciting project in the end that we were able to work on together. So thank you very much, and I'd be happy to take questions.
Yeah, thanks a lot, Trevor, for, I think, a really intriguing talk. I also really liked how you unpacked a lot of these technical details in a very accessible way. That was really great. So yeah, let me see whether there are any questions from the audience here or from folks online.
Thank you. This was really great talks. Very exciting technology. So how big so far is this repository of imaging data? I can imagine that for biologists, it'd be really useful to be able to access, a bank of control images, for example, whether it's like mice or non-human primates or humans. That would save us so much time and money in, continuously sacking these animals and restaining controls for every single experiment. So how far away are we from making that a useful repository?
That is a great question, and I think beyond my knowledge. But I would say that it's these tools are increasingly, we're in the process of writing a manuscript right now of several different, sort of the current state of next generation file formats. And there's several larger consortia efforts that are sort of all adopting the same pipelines for the data. So but a lot of these efforts are very, I'd say, like, they're not massive they're catalogs of a very specific type of data, and so it might not be sort of these all-encompassing databases. But I do think that it is increasingly, especially for newer efforts that are looking for some way of publishing their data as sort of the de facto way to approach that, maybe.
Other questions?
Sure, I think you have to. Okay. Anyways, great talk. I guess my question is sort of like, it's great to be able to search stuff up over the web, but at some point in time, locality matters, right? And so do you have a sense of where that trade-off starts to come in? You know, sort of like how much you're going to use a particular data set to bring it on to sort of local storage, because, you know, you're going to process it 100 times instead of once.
Right. I do think that these, like the cloud native architecture is really well suited for data lakes, or just sort of these long-term persistent storages that don't require super fast reads. Like, going back to the figures, like having things on the local file system is obviously is a really huge win. But I do think the next step in these file formats is we're currently investigating ways to effectively allow people to keep their data as HDF5, but also allow these remote access like similar to the indexed OMI-TIFF, these types of access patterns. And maybe there isn't actually a yes or no to what format it needs to be in, and maybe it's just you generate some extra JSON if you want to read it from the web, and that helps everything in that world, but then you also can have it in a particular environment.
Since you touched on it, I recall having seen something like HDF5 is clearly aware of the challenge, right? I thought they were introducing a new format that was, in some sense, more native to their strategies, but czar-like in the notion of having chunks that were indexed that you could pull out. Are you aware of that at all?
I don't know anything directly from the HDF5, like any specification changes, but I do know that there's a project that's called Kerchunk, which is effectively it's a tool to basically, how would I describe this? Kerchunk's a way of sort of defining that explicit referencing within metadata, and so it's sort of an additional layer that you could put on top of HDF5 that on a local file system you could read with HDF5 drivers, but then I could load this reference in my web client and then directly access the HDF5. So I haven't seen anything directly in HDF5, but I have sort of this extension, I guess. Yeah. Sure.
We have a question from Jeffrey Moffat online, who says, amazing talk, Trevor. A quick question. Sometimes in microscopy work, it is useful to create a tile of images in which there are overlaps of adjacent field of views. And Jeffrey just wonders, are there ways to combine images in SAR in a way that maintains these overlapping regions, yet continues to have all of these benefits of pyramidal representations? I don't know whether this gets too technical, but he says a single file for all FOV and many of the other features.
Yeah, that's a great question. I'm curious if so within a single array, there's no mechanism that I'm aware of to allow for these like overlapping tiles. But I would be curious if there'd be a way of representing that with multiple arrays that then you apply sort of these transformations to on the client level. And so it's really you store these separate pieces of your image separately, and then in some renderer you read those together, and then that web client performs that, or whatever the client library is, performs that alignment for you. So you save sort of the required transformations to align the two stitches together.
And then maybe one or two questions from my side. So I guess like for most practitioners, I think it will be most relevant to kind of like how do they get to a point that they can like leverage this work that is based on OMI TIFF or OMI ZAR. And if you look a little bit at like data that is also like generated here in house, like Murphy, SciSIF, so they often in their data releases have like, you know, like plain TIFFs. And how would they like get to things like OMI TIFF and like OMI ZAR? I guess they somehow need to get to some good conversion tools to write these OME headers, turn to something like bioformats. What would be the suggested strategy there?
Yeah, so I don't know for just plain TIFFs, like what the like if that's something that bioformats directly supports, but a lot of our workflow, like when we were developing the just example data sets, bioformats has these sort of two command line tools. One are bioformats to raw, and the other one's called raw to OMI TIFF.
Right.
And sort of this two-step process. And so, although I think that it might require metadata, like preemptively metadata, because that's part of the OMI conversion, is to translate that metadata to the OMI model. So, yeah, I guess I don't know for data which sort of have the metadata decoupled from the actual binary data. I'm not aware of a tool that sort of collects those, coalesces those together, and then you can run it through the traditional pipelines.
Right.
Yeah.
Okay, great. And then another question I was wondering, maybe with regard to Aviator and Vitesse. So you do all these nice modifications, selecting channels, zooming in, doing overlays and whatnot. Are there ways that you can kind of like, you know, save like a snapshot of that and kind of like reload that at a later point into the app, into Aviator?
Oh, like the current state?
Yeah, so you kind of like do all these modifications and then you arrive like at your favorite view onto this image. Can you kind of like export that in a way that you can come back on a later time point, like saving snapshots? Is this possible?
I don't believe in Aviator that's possible, but I do think in Vitesse we're working towards having like the current state of the viewer being JSON serializable. And so it's just some way that whatever state you're in, you can export that, and then you have like a reference to be able to recreate that exact view. In the Vizar viewer, actually, I'm nervous about trying to open this or trying to find it, but we've actually built in some functionality for saving the current. What am I looking for? Here we go. Some functionality for the state of the viewer. So if I go to this is an example of loading one of these OMI ZARs on the client. And if you see at the very top, the viewer state is like in the URL. So as I move around and zoom in, you'll actually see that shift. And then if I open this in a new tab, it opens to exactly to where it is in the other. So that, I think this is a very minimal example of that, but that's something that we're generally trying to apply to these other tools to allow this like linking of, oh, I saw this, and you can just share that with someone. So
yeah, so I think that would be super helpful. Okay, one more question online from Antonios, who says, great talk, thank you. Does Vizar support visualization of pointillistic data, which seems to mean single molecule localization data?
I don't believe so. I think for that type of data, I would take a look into Vitesse. Because Vitesse does support these sort of point specific data versus Vizar is very much tailored towards this like dense array data or imaging data specifically. So.
Question.
So playing, sorry, I was going to say playing off of that. Wait. Oh, sorry. Okay. I apologize for missing the first half of your talk. What I saw I really liked. Playing off of that last question, I was wondering whether there are any analogous tools for vector image data as well. And the reason I ask that is in our group, we do a lot of work with health services data, and a lot of the covariates that we line up against that are geospatially resolved. And the sort of bounding regions there are frequently just just polygons in space
Mhm.
that, think standard sort of GIS image data. Are there any analogous tools? I know this one's highly tuned specifically to rasterized images, but are there any other frameworks that you're aware of for visualizing that type of data in a browser with this sort of acceleration? I mean, just what you showed here is extremely impressive.
Like geospatial data in particular? Yeah, so the framework that we actually built all this on is based off of a geospatial library called DecGL, and they have a lot of, they support various types of like GeoJSON or Polygon data actually, so because the OMI specification doesn't have doesn't include the OMI specification doesn't include a definition for how to include vector data yet. This is something that we've sort of appended on to our Vitesse framework as well. So there's some outside of the OMI model that we allow for the definition of that type of stuff.
That's super helpful. I do think that there's a place beyond just sort of random party tricks for the image analysis, right? I think that it can be a really powerful hypothesis generation tool in a lot of different spaces. And I think the more and more of this data we're able to aggregate and sort of coalesce into the same place, I think you're going to start to see a lot more utility in those sort of visualization pipelines for other sorts of data. I mean, you guys are obviously clearly aware of the power of it for your types of data, but I think it will start to spill over into other places as well.
Would you have another question?
One quick one. We can take that offline because it's 11 now. Yeah. Was just I sort of intrigued at the start where you sort of went into, this is sort of something that sits on top of a multidimensional array, and I'm just wondering whether you've abstracted out the array representation so that some of the sort of sparse matrix formats would be easily incorporated into the sort of back end without too much work.
Yes, that's a great question. We currently have an abstraction for sort of these, like just these data sources, and the two that we support are ZAR and OMI-TIFF. And I think that we would, appending on, like sparse representations are a little bit tricky in that abstraction. Like ZAR doesn't currently have a mechanism. It's sort of, you have to have a custom serialization pipeline to be able to store sparse arrays. But I do think that if there ends up evolving that as a common standard in the bio community for saving some of these data, that will be something that we end up implementing in our clients as well. So, yeah.
Well, then, really, thanks a lot, Trevor. I think we're on time and about to be thrown out of this room very soon. So it was really a fantastic talk. I'm also closing the session on Zoom and just saying next week we will have a talk on like machine learning and clinical decision making from Brad Below-Jones. So thanks a lot, Trevor, and hope to see everyone then next week.
Yeah.
Okay.